Hi everyone, I'm Sunil Regge, consultant psychiatrist. Today I'll be taking you through the concepts of neuroplasticity. Of course, quite a prominent buzzword nowadays, a neuroessentialism buzzword. Often it's not clearly defined. So what I'm going to do is I'm going to take you through the biological, the medical understanding of neuroplasticity and what can we actually do about it? What evidence is there to improve neuroplasticity? So let's jump into it and check it out. Neuroplasticity is best defined as the ability of the nervous system to change its activity in response to intrinsic or extrinsic stimuli by reorganizing its structure, functions, or connections. The important aspect is we often think about neuroplasticity as simply beneficial. Wow, neuroplasticity should be great. That's not what neuroplasticity is, because changes can be either beneficial, which is restoration of function after an injury. It can be neutral, no change whatsoever, or negative, can have pathological consequences. So take, for example, phenomenon such as phantom limb after amputation. So individuals have an amputated limb but can still experience that the limb's there. And that happens because of a range of processes. When I show you the facets of neuroplasticity, get a better understanding as to what might happen, because one part of the brain can take over the other part's function and therefore result in phenomena such as phantom limb. Neuroplasticity is essential for learning and memory processes that allow an organism to learn and adapt to its environment. At present, there is considerable interest to harness evidence-based interventions, and we'll talk about them. And there's a massive industry from an economic, financial perspective as well to really put forward this neuroplasticity. Neuroplasticity, we can look at it from a molecular level or a cellular level. Now, at the molecular level, we have things such as signaling pathways, gene transcription, protein synthesis. At a cellular level, we can divide this into neuronal changes, dendritic changes, and synaptic changes. So synapses, of course, we have the presynaptic neuron, postsynaptic neuron, synapse right in the middle. And then we have the dendrites. As we know, the dendrites basically will occur, the presynaptic neuron, for example, something like this. When we think about the neuron, here, these are the dendrites here, right? And then we have the neuronal changes as well. Now, from a neuronal perspective, you can see there is neurogenesis. And neurogenesis, there are a number of processes such as proliferation, differentiation, survival, migration, maturation. So we'll see later that there are certain factors that are being used, potentially stem cell derived or other factors, to promote this neurogenesis. So we know things like brain-derived neurotropic factor is something that can promote neurogenesis. Similarly, dendritogenesis, which is dendritic spine morphology, dendritic spine density, changes in that. And synaptic, because at the synapse, we know that there are two processes that happen, which is the potentiation, LTP, long-term depression. And this is basically the links between neurons to other neurons. So neurons basically just a snowball effect, a firing effect, where they're transmitting information to different parts of the brain, spinal cord, etc. So these are two very important things, because we just can't have excitation or potentiation. We need the other to calm things down as well, the inhibition section. It's also divided into functional plasticity and structural plasticity. So actual structural changes or just changes occurring without any structural changes occurring. So functional plasticity, we have things such as equipotentiality, vicariation, and diaschisis. And I've talked about more in my article on neuroplasticity on the Psych Scene Hub. Certain parts of the brain can take over other parts when they're damaged. So that's where all of these equipotentiation, vicariation, diaschisis come in. So let's take the example of traumatic brain injury. We have stages of neuroplasticity, and traumatic brain injuries is well studied, so it gives us a good idea about what happens. Initially, the immediate changes are cell death occurs with a decrease in cortical inhibitory pathways for about 1 to 2 days, which recruits or unmasks new and secondary neural networks. Then there is a shift of the cortical pathway activity from inhibitory to excitatory. So initial stages is all about moving towards excitatory aspects. We know that in the brain there are neurotransmitters such as glutamate, which is excitatory, GABA, which is inhibitory. So it's really moving towards that glutamate aspect, which is important for cognition, etc. Then we have neuronal proliferation and synaptogenesis. So proliferation of neurons, synaptogenesis, basically formation of new synapses. Both neuronal and non-neuronal cells. And these are the endothelial progenitor cells, glial cells, inflammatory cells. They're all recruited to replace the damaged cells and facilitate scar tissue formation and then revascularization. So this is a process that takes a bit longer. Scar tissue, etc., revascularization for healing to occur. Then weeks after injury, new synaptic markers and axonal sprouting are upregulated, allowing for remodeling and cortical changes for recovery. Now, one of the important things here is these are some of those aspects, progenitor cells, stem cells, etc., that are used in brain injury as treatment to enhance neurogenesis and neuroplasticity. Long-lasting morphological changes occur in the hippocampus after traumatic brain injury, including the growth of the cell soma, which is really the central cell body. You know, if there's dendrites here, the central cell body is the cell soma, and recruitment of neurons to the hippocampus. Hippocampus is really where, you know, a lot of our memories reside, plus our long-term memory, implicit memory, explicit memory, etc. If you're looking to learn more about this topic and discover a range of other stimulating psychiatry courses, then don't forget to subscribe to the Academy by PsychScene. Now, what we do know is that the majority of psychiatric and neurological disorders have been associated with changes in neuroplasticity, including reductions in neurogenesis and deficits in long-term potentiation. Depression, anxiety, from a medical perspective, epilepsy, for example. Epilepsy is a great example of how seizure activity can occur aberrant synaptogenesis or aberrant neurogenesis. Furthermore, aberrant neurogenesis can also promote the destabilization of neuronal circuits and contribute to cognitive impairment. Similarly, in epilepsy, that can occur as well. And what's interesting here, from a inflammatory immune perspective, is microglia act as key modulators of neurogenesis. So initially we thought about microglia predominantly as immune cells, but they play a very important role in neurogenesis as well. So we have stress injury, they activate the microglia. The activated microglia basically, so they're resting here, they'll be activated, right? And then they move towards an immunomodulatory phase. So you can see the release of cytokines, and the immunomodulatory microglia release anti-inflammatory cytokines. So they modulate, there's this balance between inflammation and reducing inflammation. So essentially in the normal microglial resting state, the balance is tipped in the favor of neurogenesis. When activated due to stress, infection, injury, you get pro-inflammatory cytokines. So you can have a listen to the neuroinflammation video that I've done. And this enhances gliogenesis. Gliogenesis is really that scar tissue formation, the gliosis that occurs at the expense of neurogenesis. But then in the next stage, the microglia reach an immunomodulatory state where anti-inflammatory cytokines are released and the balance is once again tipped in favor of neurogenesis. So really fascinating, and a lot of this can occur both after physical trauma as well, but also stress. What are the evidence-based strategies that we know that are being used to enhance neuroplasticity, neurogenesis, neuroprotection? Cognitive training. We know that there are lots of video games, computer games, Sudoku, etc. When we look at the evidence, it's sort of somewhere in the middle. It's inconclusive. There's no definitive answer that cognitive training improves neuroplasticity. Having said that, you know, there have been studies that have looked at learning music at an early age, learning languages at an early age can be neuroprotective against the formation of dementia. They've been done there. So I guess keeping your brain active is probably a good thing overall. Then we have diet. And with diet, the two concepts I'd like to cover. One is in the book Ikigai. When they studied longevity, they found longevity was really, really high in Okinawans in Japan. And one of the things that they found was what was called hara hachibu, which means eat until you're 80% full. This was associated with improved cognitive function, happiness, longevity, etc. Kind of might be linked to the concept of intermittent fasting as well. And fasting's been something that's been there from, you know, spiritual perspective, religious perspective, where we know now that intermittent fasting, the formation of ketosis or ketones, can actually promote neuroplasticity, neurogenesis, and improve cognitive function. So intermittent fasting is likely a good thing, and hara hachibu, eating until you're 80% full, is probably a good thing. So then we know sleep. Sleep is, again, a really, really fascinating aspect overall. Sleep medicine, sleep psychiatry in its whole is a field. And you know, there are hypotheses that majority of psychiatric disorders the origin may actually lie in sleep. Recently, what they've discovered, because we sleep for a big chunk of our life. And the question is, why do we need sleep? And what they found out was, we know that sleep's necessary for consolidation of memories, learning, but it's crucial for neurogenesis, neuroprotection, neuroplasticity, because they've identified what's known as the glymphatic system, not the lymphatic system, but the glymphatic system in the brain that is responsible for the removal of toxic metabolites, toxic waste, including amyloid. Amyloid is what's deposited in Alzheimer's, associated with Alzheimer's dementia. So we've got the glymphatic system, and the glymphatic system is active in sleep, particularly during NREM sleep, deep sleep, and it facilitates the removal of all these toxic waste products, therefore promoting neuroprotection, neurogenesis, neuroplasticity. So really, really fascinating. It's important that we keep an eye on sleep overall and try to improve our non-REM or deep sleep. Then we've got neuropharmaceuticals. Now, the range of things that have been mentioned here. We have antidepressant. We have a video on antidepressants. In 2021, they identified that antidepressants may actually work through the BDNF receptor, the brain-derived neurotrophic factor receptor, which is a fertilizer for the brain, promotes neurogenesis, promotes neuroplasticity. This TRKB receptor is activated by most antidepressants, ketamine, tricyclic antidepressants, SSRIs, SNRIs, etc. So fascinating stuff. So it's not necessarily the serotonin noradrenaline that we thought about. It's likely this direct activity on BDNF, promoting proliferation, etc. So fascinating stuff. So antidepressants is one. Secondly, they've looked at things like erythropoietin. Amantadines come up, looking at promoting neurogenesis, improving cognitive function post-traumatic brain injury. Stimulants, there's some evidence that they do that post-traumatic brain injury as well. Then minocycline, antibiotic, again working through these microglia. Stem cell aspects, neurosteroids, DHEA, dehydroepiandrosterone, which is a neurosteroid in itself. So there's a lot of these aspects that are showing promise. Of course, we don't have one thing that we prescribe necessarily. It differs, and most studies are required, but there's quite a lot of medication or neuropharmaceuticals that can promote neuroplasticity. And finally, exercise. When we think about diet and exercise, these are cheap, useful ways of promoting and cognitive training, music, languages, learning languages, keeping our brain active. All of these things can be really useful ways, because the harm side is actually low, whilst the benefits may be quite significant. Exercise, we know, promotes hippocampal neurogenesis. We know reduces neuroinflammation. It releases myokines, which are anti-inflammatory, and it reduces oxidative stress. So I did a little short on exercise and how it helps the brain. Have a view of that short. So this is basically what we know about enhancing neuroplasticity. So I hope that's given you some food for thought, if you want to think about it that way, to enhance your brain function, your neuroplasticity. But I hope that it also gives you an idea about neuroplasticity being a term that really is about change in our brain to our environments. And it's not necessarily just positive. It could be positive, it could stay the same, it could be negative, and the range of things that we can do to overall provide neuroprotection to a great extent, things that are within our control. Of course, there are some other things that aren't within our control, things that obviously we need to look at from a medical perspective, holistic perspective, etc. Thank you so much for watching and giving up your time. I really appreciate it. And if you liked the video, of course, do leave us a like, subscribe to our channel, and I'll look forward to seeing you in another edition of Hub Bites. Take care and stay safe. Bye-bye. Synthesizing complex material in psychiatry to simplify your practice.